everyone, this is part 3 of the Mod 14 lecture video on molecular genetics. In part 3, we are going to be talking about the flow of genetic information from DNA to RNA to protein. We talked about this very briefly at the very beginning of the summer semester when we were talking about the main themes of biology, specifically the theme of information, but now we're going to go a lot more in depth in this. The DNA of a gene, which is a linear sequence of many nucleotides, is transcribed into RNA, which is translated into a polypeptide. And remember, polypeptides are proteins. So this graphic down here demonstrates this. We have our double-stranded DNA. Through transcription, that double-stranded DNA is transcribed into single-stranded RNA. Both of these happen in the nucleus. The RNA then leaves the nucleus and enters the cytoplasm and is translated into a protein polypeptide. So this first step, transcription, is the synthesis of RNA under the direction of DNA. And this second step, translation is the synthesis of a protein polypeptide under the direction of RNA. So DNA holds the gene, it holds the initial information. That initial information is transcribed into RNA, which carries the message outside of the nucleus and is then translated into a protein product. This is how genotype directly influences phenotype, because the genotype, the actual gene in the DNA, codes for a protein product, and those protein products influence all the functions in our body, which ultimately influence our phenotype. Genes control phenotypic traits through the expression of proteins. Currently, a gene is defined as a region of DNA that can be expressed to produce a functional product that is either a protein polypeptide or an RNA molecule. So there are some cases where the product of a gene is not a protein polypeptide. Sometimes it is an RNA molecule. Those aren't as common, but it can happen. The one gene, one enzyme hypothesis was the original hypothesis for how genes control phenotypic traits. That hypothesis was that one gene in the DNA controls the production of one enzyme. That hypothesis has since been expanded to include proteins and sometimes RNA molecules. So it's more like a one gene, one protein polypeptide, or one RNA molecule hypothesis. Researchers figured this out by working with bread mold here. They were working with bread mold strains that were deficient in one particular enzyme in a metabolic pathway, and this deficiency caused these bread molds not to be able to grow on a normal growth medium. And by manipulating the genes and making this mold deficient in different of those enzymes needed in metabolic pathways, they were able to determine which gene controlled the production of which enzyme and link the inability to grow on the growth medium back to that one gene that was not able to produce the correct enzyme. So genetic information written in codons is translated into amino acid sequences. The sequence of nucleotides in DNA provides a code for constructing a protein. So if we take a look at this image here, we have a double-stranded DNA helix with different genes. The genes are different colors. So we have gene 1 that's green, gene 2 in yellow, and then gene 3 in light blue. The gene, we're zooming in on our particular part of gene 3, and we can see the DNA sequence code right here. AAA, CCG, GCA, AAA. Transcription, which happens right here, requires the nucleic acid language of DNA up here to be rewritten or transcribed as a sequence of RNA. So it's helpful to think of transcription and translation linguistically. So when DNA is transcribed to RNA, it's still in the language of nucleic acids, but it has been rewritten in the sequence of RNA, which uses U instead of T. And then during translation, there is a change in language from the nucleotide sequence of the RNA to the amino acid sequence of the polypeptide. So in transcription, DNA is transcribed into RNA. They're still in the same language, the language of nucleic acids, but the DNA code has been rewritten in the form of RNA. And then the RNA which is in the language of nucleic acids, needs to be translated into the language of amino acids to create a protein polypeptide. And this is done using these 3-letter groups of nucleotides called codons. So a 3-letter codon codes for one particular amino acid. Experiments have verified that the flow of information from gene to protein is based on that triplet code that I just mentioned. The genetic instructions for the amino acid sequence of a polypeptide chain are written in DNA and RNA as a series of non-overlapping 3-base words called codons. So this is where those 3-base letters or 3-base words come into play. 3 RNA nucleotides will code for a specific amino acid. It took researchers a while to figure this out, but if one nucleotide coded for one amino acid, there would only be 4 possible amino acids. So we knew it couldn't be one. If 2 nucleotides coded for one amino acid, that would only give us 16 possible amino acids. And we know that there are 20 different amino acids. So researchers were able to determine that it is triplet code, so 3 nucleotide bases then code for one amino acid, which gives us more than 20 possibilities. But as you'll see in the next few slides, there is some repetitiveness where certain sequences of letters can code for the same amino acid. But just remember, these 3-base words are called codons. So UUU is a codon that codes for the square amino acid. GGC is a codon that codes for the circle amino acid. CGU is a codon that codes for the triangle amino acid. And then UUU, which we had over here, is a codon that codes for the square amino acid again. This image just shows the codons in RNA. The codons would be the same in DNA. So we would have the 3 base pairs, so AAA, CCG, GCA, and AAA. Typically, codons generally refer to RNA though, because the RNA is what is translating the information from the DNA into the amino acid sequence. The genetic code, or this triplet code that we just talked about, is the set of rules that dictates the amino acid translations of each of the mRNA nucleotide triplets. So the genetic code determines, based on the codon, the sequence of nucleotides in the codon, which amino acid is specified for. Nearly all organisms, from the smallest bacteria to large complex animals such as elephants and giant sequoia trees, all of those organisms use an identical genetic code to convert the mRNA codons transcribed from a gene to the amino acid sequence of a polypeptide. This implies that the genetic code evolved a long time ago in the history of life, because all modern life uses the same genetic code. So an early ancestor then passed this trait onto all the different branches on the tree of life. The universal genetic code is how we're able to create transgenic organisms or do genetic engineering on organisms. So this cat on the left has been genetically engineered, which just means its DNA has had a gene inserted into it that codes for a red fluorescent protein under UV light. So you can see the red fluorescent protein being expressed there. Another really common example of this, we don't do it in this class, but in other biolabs, you can take a gene from jellyfish that causes them to glow under UV light or glow in the dark, and you can, by genetic engineering, insert that gene into bacteria and create bacteria that will glow under UV light. So you're creating transgenic bacteria using a gene from jellyfish, and that works because both of those organisms have the same genetic code, so the same rules specify the translation of mRNA into an amino acid. This figure here, you'll expect to come back to this a lot. This circle basically shows you the genetic code, and this is how you figure out, based on the codon you have, what amino acid is coded for. There's also other examples of this that are squares, but I find the circular ones to be the easiest. I'm going to walk you through how to use this. So very quickly, around the outside are the names of different amino acids. These three red ones represent stop codons, so that ends translation. And then this green one here is a start codon, so that begins translation. These three inner rings are different letters that would make up the codon. So this would be the first letter in the codon. This would be the second letter in the codon, the second ring, and then the third smallest ring is the third letter in the codon. To use this, you start in the middle and work outwards. So let's say we had a sequence of U, U, G. So we're going to start here. U was our first letter. Our second letter is also U. So we're going to go to the U. Sorry, I lost my mouse. The U that is within this U's category. So U, U, and then I said G. So we're going to go to G here. That would give us leucine. Another example, let's say it's A, C, C. So we're going to start in the middle again, A, and then C, C. That gives us threonine. And then the three-letter abbreviation after is just a shorthand way to write that amino acid. I'm going to go through one example now, or one more example now. We'll have a few later on. Let's say we have CAU. So starting here, we have C, A, and then U right here would give us histidine or HIS. So that is how you use this figure to determine, based on the codon, the three mRNA base pairs, you use those to determine which amino acid is coded for. So if we look at this from DNA to RNA to protein, so we have our double-stranded DNA up here. The darker blue strand is the strand that is going to be transcribed into mRNA. So remember, to transcribe a DNA strand into RNA, you just follow the base pairing rules, but you just need to remember that RNA does not have T. Instead of T, it has U. So in RNA, A will bind with U instead of with T. So we have this DNA strand here. We're going to transcribe the top one. So T, we know it complementary base pairs with A. A will complementary base pair with U in RNA because RNA does not have T. C will base pair with G. T, A, T, A, C, G, A, U, A, U, and so on and so forth. That is how we get this RNA strand from this parent strand of DNA. And then this RNA strand will need to be split up into those three-letter codons to then determine what amino acids are coded for. So before I advance this slide so you can see the bottom, if we split this into three letters, we would have AUG is one codon, AAG is another codon, UUU is another codon, and then UAG is the final codon. So these codons are then translated into amino acids using that circle that we just looked at. So I'm going to back out of this PowerPoint so that we can go back and forth easily. So we start out with AUG. If we go look at this on our image here, we're going to start here, AUG. That gives us methionine, which is the start codon. So the start codon signals the start of translation, and it also adds the amino acid methionine to a polypeptide. So the start codon is methionine is added. Our next codon is AAG. We're going to take a look at this here, AAG. That gives us lysine or LYS, which is what they have here. Our third codon is UUU. So we're going to go back. UUU. That gives us phenylalanine or PHE, which is what they have there. Our final codon is UAG, which they're saying is a stop codon. Let's double check that. UAG, that does give us a stop codon. So that signals the end of translation. So this illustrates how information in DNA is transcribed to RNA, and then those three-letter codons in RNA are translated into amino acid polypeptides using the genetic code. Oh, don't look at the answer. So translate the RNA sequence CCA UUU ACG into the corresponding amino acid sequence. So we have CCA UUU and then ACG. I'm going to back out again. We see the answer here. Let's double check it at least. So CCA, if we go here, CCA, that gives us proline or PRO. That's what they got. The next codon, So CCA were the first 3 letters. The next codon is UUU. So we're going to go look at UUU, U, and U. That gives us phenylalanine or PHE. And then our last 3 letters are ACG. So we're going to go take a look at that. ACG. That gives us threonine or THR, which is what they got there. That is how you use the codons in RNA and translate them into the amino acid sequence. You need to use that circular figure. Now we're going to talk more in depth about the processes of transcription and translation. I just gave you a general overview of what they do. Now we're going to look more at the specific steps. So transcription, again, produces genetic messages in the form of RNA. Transcription happens in the nucleus. So in the nucleus, the DNA helix will unzip, RNA nucleotides will line up, and RNA polymerase will join them along one strand of the DNA following the base pairing rules. So remember, transcription is transcribing the nucleic acid sequence of DNA to the nucleic acid sequence of RNA. When we are talking about DNA synthesis or DNA replication, the enzyme that facilitated DNA replication was DNA polymerase. Now we're talking about transcription, so transcribing DNA to RNA. The enzyme that facilitates that is RNA polymerase. So RNA polymerase joins the nucleotides along one strand of DNA following the base pairing rules. A specific nucleotide sequence called a promoter, shown right here, acts as a binding site for RNA polymerase and determines where transcription starts. So a gene needs a promoter in order for transcription to start. If we take a look at this figure, we have the DNA double helix here. We have the RNA polymerase enzyme coming in. It will bind to the promoter. The DNA will start to unwind and only one strand will be used as the template. The other strand will be unused. The template strand is what RNA polymerase will use to create the complementary RNA strand. RNA polymerase will continue to add RNA nucleotides until it reaches a sequence of DNA bases called the terminator. The terminator signals the end of the gene. The terminator is over here. So the promoter in green signals the beginning of transcription, and then the terminator over here in red signals the end of transcription. So RNA polymerase here is going to continue adding free RNA nucleotides that are complementary to the template strand. Remember, the other strand is unused. And then after the template strand has already been transcribed into RNA, the DNA will start to rewind. And then in termination, once RNA polymerase reaches the terminator, it's going to break off from the DNA and the completed RNA will break off from RNA polymerase. So this is an overall overview of transcription. Transcription takes place in 3 phases. We have initiation, which is the first phase, elongation, which is the second phase, and then termination, which is the third phase. In initiation, RNA synthesis begins after RNA polymerase attaches to the promoter, like we already talked about. DNA will unwind and then RNA will use the template strand to create RNA polymerase will use the template strand to create a new single-stranded RNA. The second stage of transcription is elongation. That's just when the newly formed RNA strand grows as RNA polymerase continues adding new nucleotides. And then finally, the third stage is termination, which is when RNA synthesis ends, when RNA polymerase reaches the terminator DNA sequence and the completed RNA is released. So those are the 3 steps of transcription. Here's a quick animation video on the process of transcription.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that's transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
So that is the process of transcription. A quick checkpoint question based on everything we just talked about. How does RNA polymerase recognize the start and the end of a gene? Take a second to think about that. Hopefully you said that the promoter indicates the start of the gene and then the terminator indicates the end of the gene. Special DNA sequences mark the start and the end of a gene. Those are the promoter and the terminator. Now we have our RNA that has been produced. That eukaryotic RNA is processed before leaving the nucleus as mRNA or messenger RNA. So the kind of RNA that encodes amino acid sequences is called messenger RNA or mRNA. You'll hear me refer to it too, because it conveys genetic messages from DNA to the translation machinery of the cell. So messenger RNA is what leaves the nucleus through the nuclear pores and then goes to the ribosomes for translation to occur. Once you have your RNA transcript or your RNA strand that has been produced from transcription, that RNA has to be processed until it is considered messenger RNA. So before leaving the nucleus as messenger RNA or mRNA, eukaryotic transcripts undergo RNA splicing. And in RNA splicing, a few different things happen. The first is that introns, which are non-coding segments of RNA, are spliced out. So within the DNA of all organisms, we have large stretches of introns, which are non-coding DNA. Scientists still don't know quite what they're for, but they are important, but they don't code for RNA and protein products. So introns are the same in RNA. They're non-coding segments of RNA. They don't code for anything, so they need to be cut out of the RNA transcript. So this lighter blue here is an intron, and this lighter blue is an intron in DNA. When that DNA is transcribed into RNA, the intron is shown in light pink. The first thing that needs to happen is those introns need to be removed, so they're just kind of cut out. Exons are the part of a gene that are expressed. In DNA, they're shown in darker blue here, and then in RNA, the exons are in the darker pink. The exons are then spliced together once the introns have been removed. So the introns, the light pink, are taken out. The dark pink sections need to then be attached together. And then the last thing that happens is a cap and a tail are added to the ends of the mRNA. And this actually happens up here in this image. We have the yellow cap there and then the yellow tail at the end. The cap and the tail are important so that the mRNA doesn't degrade. They kind of help protect the important coding part of the mRNA, the coding sequence. Once all of that has happened, so once the introns are spliced out, the exons are spliced together, and the cap and the tail have been added, the mRNA is now ready to leave the nucleus through the nuclear pores. And so that's just a bigger image of what we just talked about. A checkpoint question based on this: explain why most eukaryotic genes are longer than the mRNA that leaves the nucleus. We just talked about this, but the genes have introns in them, which are the non-coding regions of the gene. The non-coding regions are cut out or spliced out in the mRNA, which is why the mRNA is shorter than the eukaryotic gene. So their official answer was these genes have introns, which are non-coding sequences of nucleotides that are spliced out of the initial RNA transcript to produce mRNA, messenger RNA. Now we're going to talk more about translation. So transcription takes place in the nucleus in eukaryotic cells, and then translation takes place in the cytoplasm using ribosomes, because remember, the ribosomes make proteins. Specifically, a ribosome attaches to the mRNA and translates its message into a specific polypeptide, a protein polypeptide. And it does this aided by transfer RNAs or tRNAs. So there's a few different types of RNAs that we're going to talk about. We already talked about mRNAs, which are the messenger RNAs, which carry the message from DNA to RNA to the ribosomes for protein translation. tRNAs are transfer RNAs, and transfer RNAs help in translation from mRNA to amino acid sequences. Each transfer RNA or tRNA is a folded molecule bearing a base triplet called an anticodon on one end and a specific amino acid attachment site at the other end. Down here are 3 different drawing representations of a tRNA. The anticodon is down here in this image, here in this image, and then down here. And then the amino acid attachment site is shown here in purple. Transfer RNAs have these chemically modified bases represented by asterisks. We don't quite know what they do, but we know they're important because the transfer RNAs don't function without them. The anticodon down here is what will recognize the specific codon in the mRNA. And then the amino acid attachment site is where this particular transfer RNA's amino acid attaches. Each transfer RNA, depending on its anticodon down here, will only carry one specific type of amino acid. These little dashed lines are hydrogen bonds, just showing the folded polynucleotide structure of the transfer RNA. And then this image on the right is a simplified representation of a tRNA. This is the one we'll kind of use going forward. Acid attaches up here, and then the anticodon is down here. Remember, the anticodon is what will recognize the codon in mRNA. And this is a bigger image of the transfer RNAs. Each transfer RNA, based on its anticodon, will bind to one particular type of amino acid. And the tRNA binding to the amino acid is facilitated by a specific enzyme. You'll see the enzyme here in blue. There is one specific enzyme for each amino acid, a different enzyme for each amino acid that facilitates the bonding of that particular amino acid to its specific tRNA. So like phenylalanine has its own enzyme that will bind it to tRNA. Leucine has its own enzyme that will bind it to tRNA, etc. The protein polypeptides are actually built in the ribosomes. When we talked about the tour of the cell, we talked about how ribosomes are where proteins are made. We're going to look at the ribosome more in depth now. Ribosomes are structures in the cytoplasm that coordinate functioning of mRNA, messenger RNA, and tRNA, transfer RNA, and catalyze the synthesis of protein polypeptides. Ribosomes are made of ribosomal RNA, rRNA, so that's another type of letter RNA. Ribosomal RNA or rRNA helps make ribosomes along with proteins. And the ribosomes have binding sites for both the transfer RNAs and the messenger RNA. Remember, ribosomes have a large subunit and a small subunit, and this image actually shows the mRNA, the tRNA molecules, and the growing protein polypeptide that is starting to extend from the ribosome. If we take a closer look at the ribosomes, the small subunit down here is where the ribosome attaches to the mRNA. There's an mRNA binding site on the small subunit. The large subunit is where the tRNA binding sites are. So there's 2 tRNA binding sites, the P site and the A site. tRNA can bind to either of those, and then the amino acid polypeptide and the transfer RNAs can exit the large subunit through this exit tunnel up here. If we take a look at the visual of this with mRNA and tRNA, we see the largest subunit of the ribosome up here, the small subunit down here. The mRNA is bound to the small subunit, and then the large subunit has bound to tRNA molecules. The growing polypeptide exits via the exit tunnel. And we can see here the next transfer RNA has already bound to its complementary mRNA, and it's carrying the next amino acid that needs to be added to the polypeptide. The tRNAs recognize the mRNA sequence that they need to bind to through their anticodons, which are the complementary sequence to the 3-letter codon in the mRNA. An initiation codon marks the start of an mRNA message. Translation can be divided into the same 3 phases as transcription. We have initiation, elongation, and finally termination. So we see the cap in this mRNA molecule and the tail, which just help protect the molecule from degradation. The actual genetic message, the actual codons, are in dark pink. At the beginning and at the end, there are these regions that are non-coding. They're initiation codons. So they mark the start of the mRNA message, and they help facilitate the binding of the mRNA to that small subunit of the ribosome. Initiation, which is the first step of translation. Initiation brings together mRNA, a tRNA bearing the first amino acid, and the two subunits of a ribosome. And initiation occurs in two steps, which are shown below. In the first step, we only have the small ribosomal subunit that has assembled. The mRNA see this non-coding sequence here that helps facilitate the binding. The mRNA has bound to the small subunit, and then the actual genetic sequence, the codons start with the dark pink. So mRNA has bound to the small ribosomal subunit, and then the initiator tRNA, which is a start codon and carries methionine as the amino acid, has bound to the start codon in the mRNA. So AUG is a start codon. It codes for methionine and that green light in the circle. The initiator tRNA knows where to bind because its anticodon, shown in green, is complementary to the start codon AUG. So A binds with U, U binds with A, and then G binds with C. So it's complementary. That's how it knows where to bind, and it is carrying methionine. The second step is the large ribosomal subunit will now attach to this structure that we had over here. And that initiator tRNA carrying the methionine will go into the large ribosome's P site. Now the A site is open for the next transfer RNA to come into the large ribosomal subunit. So a checkpoint question: what would happen if a genetic mutation in a gene changed the start codon to some other codon? So if we didn't have the start codon, the initiation of translation would not occur. So the messenger RNA transcribed from the mutated gene would be non-functional because ribosomes could not initiate translation correctly without that start codon. The start codon is critical. The next step is elongation, and elongation adds amino acids to the polypeptide chain until a stop codon terminates translation. As the mRNA moves one codon at a time relative to the ribosome, a tRNA with a complementary anticodon pairs with each codon, adding its amino acid to the growing polypeptide chain. Elongation continues until a stop codon reaches the ribosome's A site, which is this site over here. So this is a written out explanation of what happens. And then over here on the right, we have a drawing of what happens. I have this a little larger on the next few slides, so I'm going to go ahead and advance the slide and talk through this drawing, which is showing what I just read. So we start out with the ribosome assembled. We have the large subunit and the small subunit. Methionine this polypeptide has grown a little bit, so this is probably not methionine anymore. It's another tRNA. The first step in this sequence is that the anticodon on a transfer RNA is going to recognize its complementary sequence, its complementary codon in the mRNA, and then it will attach at the ribosome's A site. So this anticodon matches this codon. So now that tRNA is going to attach to the A site. Once that tRNA with the square amino acid has attached to the A site, a peptide bond is going to form between this growing polypeptide chain and the new polypeptide here. So this circle polypeptide is currently bound to this tRNA. It's going to break that bond and form a peptide bond to the square. So it will now be attached to the square and its tRNA. This tRNA will not be attached to anything. The third step, so we see that peptide bond formation going on here. The third step is translocation. So this tRNA that was here that is no longer attached to anything leaves the ribosome. Now we have this tRNA that was holding the square polypeptide attached to the growing polypeptide. This transfer RNA is going to translocate over to the P site on the left, which will leave the A site open. So translocation occurs, this tRNA jumps over here, and now this A site is available for the next tRNA that is complementary to this codon here to come in and bind and then attach its amino acid. So I'm going to talk through that one more time a little quicker. We have the growing polypeptide here, the P site and the A site. A new tRNA that has a complementary anticodon to this codon in mRNA will come bind. Once this tRNA has come and bound to the ribosome's A site, a peptide bond will then form between this growing polypeptide chain and the new amino acid, so between the circle and the square here. Once the peptide bond forms, this tRNA will no longer be attached to anything, so it will translocate outside of the ribosome, and then this tRNA on the right will now be attached to the growing polypeptide chain, which we see right here. This tRNA will then translocate to the P site. Once it is in the P site, the A site is now available for this whole process to start over and for the next tRNA with a complementary anticodon to this codon in mRNA to come bind. So checkpoint question, what would happen if a mutation caused a codon in the middle of an mRNA to change from UUA to UAA? So we got to look at what UUA is and then what UAA is. So go back to our circle. So UUA codes for leucine, but then if we have UAA, that codes for a stop codon. So if that happened, let's go back to the checkpoint question. If that were to occur, translation would stop prematurely because a stop codon was introduced. So the whole rest of the polypeptide would not be translated because there was a premature start codon. Okay, so these next few slides are a review of everything we've talked about in this video. So the sequence of codons in DNA via the sequence of codons in mRNA spells out the primary structure of a polypeptide, a protein polypeptide. We're going to walk through all of those steps again briefly. So the first step is transcription, where DNA is transcribed from where RNA is transcribed from DNA. So transcription, mRNA is transcribed from the DNA template. RNA polymerase facilitates transcription and it will create a messenger RNA, single-stranded RNA. That happens in the nucleus. That mRNA will then be modified and exit the nucleus through the nuclear pore, and translation will take place in the cytoplasm. The second step here is the amino acid attachment. Each amino acid attaches to its proper tRNA with the help of a specific enzyme in ATP. That was the one image we looked at. Each amino acid has its own specific enzyme that will attach it to its tRNA. The third step is initiation of polypeptide synthesis. The mRNA in pink, the first tRNA, which carries the start codon, methionine, and then the ribosomal subunits come together. The fourth step is elongation. So a succession of tRNAs add their amino acids to the polypeptide chain as the mRNA is moved through the ribosome, one codon at a time. And then finally, termination. The ribosome recognizes a stop codon. The polypeptide is terminated and then released from the ribosome, where it will assume its folded polypeptide shape. Which of the types of nucleic acids you've learned about does not participate directly in translation? So if we just take a brief look at this figure, we know RNA participates directly in translation because the mRNA strand is the template for the tRNAs, but DNA never leaves the nucleus, so DNA does not participate directly in translation. DNA is in the nucleus where transcription occurs, but it's RNA, specifically messenger RNA, that takes that message to the ribosomes. The last thing we're going to talk about in this video are mutations. Mutations are changes in the genetic information of a cell or virus caused by errors in DNA replication or recombination, or by physical or chemical agents called mutagens. So DNA is normally very precise when it replicates, but errors can occur. And then physical or chemical agents called mutagens can also lead to mutations in the DNA. Some examples of mutagens are cigarette smoke, UV light from the sun, things like that. There are different types of mutations, and each one has different effects. So substituting, inserting, or deleting nucleotides alters a gene with varying effects. This image here shows the single mutation that causes sickle cell anemia. So in the gene for hemoglobin, the normal hemoglobin DNA has the sequence CTT. The complementary mRNA strand to that is GAA. GAA codes for the amino acid GLU or glutamine, and that having glutamine there produces normal hemoglobin. In someone with sickle cell anemia, they just have one nucleotide substitution here. So instead of CTT, they have CAT. The resulting mRNA from that is GUA. So there's a U here instead of an A. As a result, GUA calls for valine instead of glutamine, and valine here instead of glutamine results in sickle-celled hemoglobin that crystallizes. So just one mutation can have pretty serious effects in the body. I want to go out to the whiteboard now and talk about a few different types of mutations. So the mutation we just saw an example of was a nucleotide substitution, where one nucleotide was substituted and that resulted in a different amino acid. That's an example of a missense mutation. So a missense mutation is a nucleotide substitution, but it's a nucleotide substitution that calls for a different amino acid. So it called for valine instead of glutamine. So I'm going to write out nucleotide substitution here. And so a missense mutation is a nucleotide substitution that calls for a different amino acid. We're going to jump over here to the left. A silent mutation is also a nucleotide substitution, but a silent mutation still codes for the same amino acid. And this is because on that circle diagram where you figure out what amino acid is called for, I'm going to go ahead and pull it up. There are multiple ways to get the same amino acid. So like if we look at serine, there are multiple different ways to produce serine. You could have UCA, UCC, UCU, all of those would produce serine. And then over here, serine shows up again. So AGC and AGU would also produce serine. So there can be situations when a nucleotide substitution doesn't really do anything at all. It still codes for the same amino acid. That's known as a silent mutation, and that is caused by nucleotide substitution. We're going to jump over here now. A nonsense mutation is when a codon is changed to a stop codon. Again, this is caused by a nucleotide substitution. Nonsense mutations are generally not good because they're just going to stop translation right where that stop codon occurs. So it's going to end up with a prematurely terminated polypeptide. And a nucleotide substitution causes this. So likely the polypeptide will not be functional. The last type of mutation is a frame shift mutation. A frame shift mutation is when there is a nucleotide insertion or deletion. So a nucleotide is an insertion is when a nucleotide is added, and then a nucleotide deletion is when a nucleotide is taken away. Frame shift mutations alter the reading frame, so they're generally very detrimental to the protein polypeptide product. So those are our 4 types of mutations: silent, missense, nonsense, and frame shift mutations. We're going to walk through a few examples of each. So we have a normal DNA gene on the top and then the normal mRNA on the bottom, followed by the normal protein sequence. A nucleotide substitution here, this is going to be a missense mutation because it will substitute a nucleotide, a single nucleotide in the mRNA, which results in a different nucleic acid. So we have serine instead of glycine here because this mRNA should have been a G, but instead it is an A. So we get serine. A nucleotide deletion is going to mess up the whole reading frame after the deletion because this U right here is just deleted. It just got booted. So instead of phenylalanine here, UUU, it now is UUG, which codes for leucine, and then it's GCG, which codes for alanine, and then the CA don't even have a third codon. Same with the nucleotide insertion. So it's supposed to be UUU, but a G has been inserted here. This then throws off the whole reading frame after the insertion because now UUG codes for leucine, UGG codes for tryptophan, and then CGC codes for arginine. So it throws off the whole reading frame. To help conceptualize this, I've done mutations in a few sentences so that you can see the difference one single letter makes. So we're going to only make sentences with 3-letter words to represent the triplet codons. We're going to start with the sentence, the cat ate the rat. So all 3-letter words. We're then going to do a nucleotide substitution and change one letter. The R in rat is going to change to an H. So now our sentence reads, the cat ate the hat. It's still a readable sentence, but did we change the meaning of the sentence? Yes, we did, because now the cat is eating a hat instead of a rat. Now I want to show you what happens with the frameshift mutations. So a nucleotide insertion or a nucleotide deletion. So we're going to add a letter B in the 6th letter place. So now our sentence reads, the cab tat eth era T. Does that make any sense at all? No. So by adding a nucleotide insertion, adding the letter B here, the whole rest of the nucleotide sequence has now become nonsense. It doesn't make sense at all. And the same thing happens if we have a nucleotide deletion. So if a letter is deleted and everything is moved over, we're going to delete the letter C and move everything over. Now we have the ada, tet, her, at. So again, the sentence is now basically nonsense. It doesn't mean anything. And this is why those frameshift mutations, the nucleotide insertions or the nucleotide deletions are so detrimental because they mess up the entire rest of the reading frame, the rest of the sentence after you get a nucleotide insertion or a nucleotide deletion. So our final checkpoint question, how could a single nucleotide substitution result in a shortened protein product? Take a second and think about this, and then we'll come back together. So if you said a substitution that changed an amino acid codon into a stop codon would produce a prematurely terminated polypeptide, you would be correct. These premature stop codons are also very detrimental because the protein is going to be nowhere near functional. So this is the end of the lecture on molecular genetics. I'll see you guys again for the next module on regulation of gene expression.